Thanks for tuning in to First Principles Daily, Episode 18. It's June 23rd, 2026. Today's question: what would this cost if you only paid for the atoms? The first human genome cost billions and took 13 years. Today, the same sequence costs a few $100s in hours because someone treated DNA as millions of parallel reactions instead of one at a time. The Human Genome Project delivered one complete sequence at a reported cost on the order of $3 billion after 13 years of work. Today, a whole human genome can be sequenced for a few $100s in well under a day. The difference did not come from faster Sanger machines or cheaper labor. It came from discarding the single reaction model entirely and rebuilding the process around the physical fact that millions of DNA fragments can be read simultaneously on one surface. Sanger sequencing had been the established method since the late 1970s. Laboratories scaled it by running more capillary tubes in parallel and by automating the fluorescent readout. Yet the core chemistry remained one template molecule processed at a time, inside one reaction volume. Each base call required its own enzymatic extension, termination, and separation step. Throughput therefore scaled roughly with the number of capillaries or lanes an instrument could hold. The finished cost reflected that serial limit. Reagents, enzymes, and instrument time were purchased per reaction. The price per base stayed high even after incremental automation. The convention felt natural because every prior molecular biology workflow had also been serial. Nobody questioned whether the physics of DNA itself demanded one molecule at a time. As a result, the early 2000s price for a mammalian-sized genome remained in the tens of millions of dollars when estimated by extrapolating Sanger runs. The field accepted the cost because the method had always worked that way. Regulatory and grant structures rewarded incremental improvements over wholesale redesign. The decisive break came when engineers asked what the raw materials alone would cost if every DNA fragment could be read at once. A rough magic wand estimate begins with the consumables required for one flow cell. That estimate starts with a few dollars worth of nucleotides. It adds perhaps $10 to $20 in polymerase and other enzymes. It includes another $10 to $15 for the surface chemistry and optical or semiconductor detection layer. Adding buffer salts, wash solutions, and minor plastics brings the total raw material floor to roughly $30 to $60 per high output run. The older Sanger-based estimate carried an idiot index of several $100,000 because the finished price sat orders of magnitude above that floor. The idiot index measures how many times more the finished consumables cost than the raw materials inside them. Under Sanger, the multiplier reached several $100,000 because each serial reaction paid full price for enzymes and separation hardware that the raw atoms alone would never require. The first redesign move was to immobilize millions of separate DNA fragments on a single planar surface. The same polymerase and nucleotide mix could then act on all of them simultaneously. The chemistry itself did not change, only the spatial arrangement. The second move replaced the serial efflector-effectorate separation of Sanger with cyclic addition of reversible terminators whose fluorescence could be imaged in place. This eliminated the need for a separate separation step per fragment. The third move shrank reaction volumes from microliters to picoliters by confining each cluster inside a tiny well or by using bridge amplification on the surface itself. Reagent cost therefore fell in proportion to volume. On some later platforms, a fourth move replaced optical cameras with semiconductor sensors built directly under the flow cell. This removed the expensive objective lenses and alignment hardware while preserving base calling accuracy. Each of these steps attacked a different term in the cost equation. Parallelism, separation, volume, and detection were addressed rather than optimizing the legacy instrument. The key engineering trade-off at every stage was maintaining sufficient signal-to-noise so that the error rate did not rise faster than the throughput gain. That requirement was met by increasing cluster density and by refining the chemistry of reversible terminators until raw accuracy approached Sanger levels. The net result is that a production-scale instrument now delivers a 30-fold coverage human genome for a few $100s in consumables and roughly 1 day of runtime. The new idiot index sits somewhere between 5 and 15. It remains well above the physical floor, but sits orders of magnitude lower than the Sanger baseline. The remaining gap lives mostly in the cost of interpretation, data storage, and clinical validation rather than in the sequencing chemistry itself. Parallel sequencing also introduced new limits. Short reads complicate assembly of repetitive regions. Higher raw error rates must be corrected by deeper coverage. The sheer volume of data now dominates downstream analysis budgets. Those constraints are genuine and have not been removed by the cost collapse. They simply moved the bottleneck from base calling to meaning making. When a finished process costs many times its raw materials, the design or the workflow is the problem, not the scarcity of atoms. The magic wand floor shows that the atoms themselves are not the expensive part. The several 100,000-fold idiot index under Sanger arose because every step assumed one reaction volume at a time. Once the fragments were allowed to sit side by side on a surface, the same reagent served millions of templates instead of one. Volume reduction then multiplied the savings because picoliter wells replaced microliter tubes. Imaging in place removed the electrophoresis hardware that had scaled linearly with each additional base call. Semiconductor integration later cut the optical overhead that had remained even after parallelism arrived. Each change lowered a distinct cost driver while the underlying chemistry stayed intact. Questioning whether a reaction must stay serial is often the highest leverage first step. The same move, turning a serial operation into millions of simultaneous ones, has already appeared in solar cell metallization, battery electrode coating, and messenger RNA manufacturing. In each case, the raw material floor stayed roughly constant while the finished price dropped because the design stopped paying for unnecessary separation steps and oversized reaction vessels. The pattern suggests that any laboratory or manufacturing sequence whose price greatly exceeds its atoms is still organized around an assumption that one unit must be handled at a time. That assumption can be tested directly by asking how the process would look if every molecule or part could interact with the reagents at once. The genome example shows the arithmetic works when the physics permits it. Which other entrenched laboratory or manufacturing step still assumes one molecule or one part at a time when the physics allows otherwise? That's a wrap on today's First Principles breakdown. The ideas travel further when you pass them on. Send this to a fellow builder. I'm Patrick in Vancouver. See you tomorrow. And if you'd rather watch than listen, find us on YouTube at Nerra Network. Links in the show notes. This episode used AI voice synthesis of my voice. Editorial selection and analysis are my own.